Okay, thank you very much. I would like to thank the organizers for the opportunity to speak in this meeting. I'd like to speak about some work we did on adipose tissue, and that's actually a work involving a certain number of people. So first is Diane Perichard, who is in this room, who was a PhD student in Toulouse and Imperial College, and Fanny Delbecq. Then there is a team of biologists from this laboratory, Stroma Lab in Toulouse, who's, as the name indicates, a laboratory which specifically studies the stroma, the mesenchymal tissues. And then there is an experimentalist doing the, actually this is an imaging platform, and some computer scientists that help us in the imaging. And we've been sponsored by various bodies here. All right, so yeah, so let me start with the problem and sort of a kind of a small literature review. So the overall goal is try to explore possible mechanisms of tissue self-organization and homeostasis. And the motivations are tissue regenerations, tissue engineering, and the study of tissue homeostasis disorders like fibrosis, inflammation, and so on. So the Stroma Lab laboratory is a laboratory which is strongly involved in regenerative medicine. It's actually co-sponsored by the French Blood Institute, which is one of the major players in France about regenerative medicine. And they like to actually study and devise cures involving tissue regeneration, not only tissue engineering, meaning that you kind of produce a tissue, artificial tissue in an external scaffold, for instance, by seeding some stem cells and letting the tissue develop and reimplanting the tissue, the organ in the body after the organ has grown outside the body, but rather really trying to trigger, metabolic changes in the body such that the organ that has been sort of injured is grown again in its in previous shape. It's in operational state. Many often when you have healing, then that regenerates a organ but with reduced functionalities. And the problem is really to try to see how to convert or to switch back to a sort of a pathway that actually would allow the regrowth of the tissue in its original functionality. So that's the overall goal. It's very ambitious, and so we are not obviously not doing this because otherwise we would be famous. But so in this laboratory they use adipose tissue as a model for this. Not that there are many interesting issues related to adipose tissue per se, which are related to health issues like obesity. Obesity is a very actually it's an epidemic. It's probably the biggest epidemic in the world nowadays. Okay, so there was a poll in the 2007 or 2009 in UK that more than half of the population is overweight or obese. So it's really a very and it's by the lifestyle, that's a Western lifestyle that expanding to developing countries. That's actually there is a prospect that this epidemic is going to grow even further. So it's a very important problem. Obesity has various consequences on vascular diseases, cancer, and so on. And so there is actually probably compared to the stakes not enough work done on this, but there are other issues related to the adipose tissue. It is an easily accessible organ. It's just under the skin. So if you can actually use that organ to produce or to sort of as a kind of machinery to help you produce other organs, that would be interesting in regenerative medicine. It turns out that adipose tissue has a lot of functionalities. It's not just stupid fat. It has a lot of functionalities like immunity. It has a lot of stem cells and so on. And actually, these stem cells are quite pluripotent, so it's actually able to actually generate other things than just adipose tissue. So there's a huge interest in using adipose tissue in regenerative medicine. Yeah, it's also actually the organization of the adipose tissue is actually representative of the organization of many other organs. So as we see here in this picture, that so the adipocytes, so these are the cells, the functional cells of the adipose tissue, which are full of fat, have been stained in red, and the collagen matrix is in blue, and in green you can see actually the staining doesn't distinguish between the stem cells and the capillaries, the blood vessels, but you can see that there is an organization, as a rather well-defined organization of collagen fibers surrounding groupments of cells in the form of, roughly speaking, a rugby ball, okay, at least in this 2D image. And that's actually this organization into lobules, into functional subunits of organs is actually a rather, I mean, the morphologies are different from organ to organ, but there is a lot of organization of organs into these functional subunits like this. So looking at the organization of the adipose tissue is actually interesting because it helps in understanding organization of other types of organs. Okay, so
what's the scale here?
I don't remember. I think that's 100 microns, something like that. So yeah, so it should be like that. Yeah. Okay, so we would like to investigate the mechanism by which the lobular organization takes place and is maintained. Okay, so this is basically the main problem. And okay, we worked out from a very simple hypothesis, which is actually very similar to the one that was worked out in the previous talk. That's actually mostly mechanical interaction that drives the organization of the tissue. And the idea is that there is a scenario where you have collagen fibers resist to bending. So when you have adipocytes that grow, then they bend the collagen fibers. But on the other hand, this bending resistance actually forces new adipocytes to regroup with existing adipocytes. And then the growing clusters of adipocytes actually force the fibers themselves to regroup into thin, into elongated structures like that. So that was basically the idea that we had. We tried to see if it could be valid. Okay, so we used as a methodology, we started with an individual-based model for cells and fiber elements, which I'm going to develop. We have also developed from this a continuum models for fibers alone so far. We are planning to actually develop models also for the cells and coupling them. And so before going to what we did, I'm going to try to make a short literature review, basically centered on the individual-based model, because I will center this talk on the individual-based model. I will briefly mention the continuum model at the end of the talk, but I thought that due to time constraints, it was better to focus on only one model. Okay, so in the literature there are many models, and I'm pretty sure I've forgotten many people and many works, and so I try to do my best. But if I missed some important references, I will be very happy to that you pointed to me. There are a lot of literature about cell-based models, so essentially you're following cells. There is a lot of literature also on fiber models, and then there is some literature on coupled cell and fiber models. So the most, I think the most important, I mean, the largest literature is about cell-based models. So so basically, I try to classify them into two big classes. There are models where the cells are really simple objects like spheres, okay, and where, generally speaking, the non-overlapping of the cells is taken care of by a repulsing force. And actually, there is also a cell attachment, which is actually taken care of by an attractive force. So there is actually the potential is kind of short-range repulsive, large-range attractive. And usually these are off-lattice models, and I've given a couple of names here. I think that picture is taken from the paper by Bayer, where there is a lot of, it's actually very complex models involving many agents. So involving actually epithelial mesenchymal transition in cancer. But in many cases, cells are models that are more complex objects because they have a certain, we want to describe a certain shape. So in this area, there are various ideas. There are lattice-based models, which are actually called cellular pots models. Here is a picture of paper by Merckx and Glaser, where you can see actually each cell is actually described as on a lattice, but as a complex object. There is a kind of off-lattice version of that, which is called the subcellular element model, which has been developed by Tim Newman in Dundee, and it actually produces very nice pictures of, like, for instance, a tumor spheroid, where you can see the cells and the sub-elements. There's been a very popular model also, this is called the vertex model, which is basically targeted as in epithelial tissues, where you really like to model very precisely the attachments of the cells with the nearby ones, and you actually want to monitor how faces these polygons have, and how this number of faces evolve in the course of division and evolution of the tissue. Actually, these models have are quite nice in 2D. When it comes to 3D, it becomes really complicated because you have polygons in 3D. So there are some teams that are developing 3D vertex models, but it involves a lot of engineering. And so we actually, just to position our work, we're going to actually try to be as simple as possible. So we would rather belong to the first category. Now for fibers, there's been a lot of work on cross-link or networks of cross-link fibers or equivalently lattice spring models, and in particular related to the description of the actin network in the lamellipodia, for instance. So I have cited a certain number of papers here. So basically, you're modeling, really the mechanics of the fiber. Each element is some kind of a beam which can stretch, bend, or undergo torsion, and then you connect these. You can measure various things, and eventually you measure kind of lattice elastic constants or things like that. There's been actually an interesting work involving phase transition between bending and stretching. So roughly speaking, when you have a matrix, and if you say, for instance, if you stretch it, or if you stretch it, or if you shear it, then at low fiber density, then basically the medium is going to resist by bending. Whereas if you have a large number of fibers, then it's rather stretching that gets involved, and there is a phase transition between bending and stretching that has been involved. Recently, there's been a model more inspired by flocking models, and actually bears some kind of similarity with the method that we saw in the last talk, where the interaction is computed by some kind of neighboring of averaging over the neighbors. So that's this fiber flocking model by Anzo and co-authors. And we're also going to get inspired by this kind of approach. All right. So now what's happening for coupling cells and fibers, then there is much less work that I know of. There is one series of work by Hillen and Painter and others where they have modeled the motion of that's basically what's called the cell crawling. So cell moving along the direction of the fibers and simultaneously remodeling the network. So, for instance, cells are gonna sort of cut the fibers that are across their way and favor the remodeling of the matrix with fibers in the direction of their motion, and that obviously favors then the motion of consecutive cells in chains like this. Okay, so there's been modeled like this. But in this case, the feedback between the cells and the fibers is more like chemical feedback. There is, proteolysis that the cell really, changes the nature of the fibers by chemical interaction. And I don't think that in this work there was any consideration of the mechanical feedback. So in this present work, we were rather explore the mechanical feedbacks and saying that there is the resistance of the fibers to bending and the resistance of cells to compression. So that's basically the ingredients of our mechanical feedback. Actually, in this case, there is literature about that, but this is away from, mostly away from biology. For instance, there's been work by physicists where mixing steel rods with steel spheres and showing that by reducing the available space, the steel spheres force the steel rods to align. Okay, and there's been also a work which has been done in actin and with some polymer beads, which show a similar effect. And the idea is that this is really an effect which is due to what has been actually described in terms of what's called the depletion force, which is really something that's related to noise. The idea is that because of volume exclusion, it's entropically or free energetically favorable for the rods to align like this, right? Because if there is too little space between them, then the steel spheres cannot penetrate in the interspace here, and because they are bumping, basically because of noise on the other side, they actually force the steel rods to align. So here we are not going to consider noise as a major player, but what generates this similar kind of force is actually some kind of contact repulsion between the fibers and the cell, which plays a little bit the same role. And here what we want to really see is not only an anisotropic effect on the fibers, but also on the clusters of spherical elements, which we do not really see here. So that's actually some kind of novel effect that to my knowledge, hasn't been explored here. Okay, so that's the thing. Yeah, so in general, what we found is that in the literature review, the biology is very detailed. That's good, because if you are looking for a quantitative agreement, of course, you need to be precise in the modeling. On the other hand, if there are too many ingredients, then it's very difficult to disentangle the various phenomena, and in particular to know which one is important, which one is less important, and so on. And there is another thing that we'd like to do is actually use this individual-based model to derive a macroscopic model, because we think that it's important that the macroscopic models and the microscopic models are really cross-linked. Because the future, I think, is a kind of hybrid modeling where you will have some parts of the system which is microscopically described and other parts which are macroscopically described. And if you're using two different models that have, you know, relations with each other which is not clear, then it's not very, at least intellectually, very satisfactory. So we would like to be able to derive macroscopic models, and in order to derive them as rigorously as possible, then it's desirable to start from a very simple, as simple as possible microscopic model. So we are going to sort of abandon the idea of being quantitatively accurate, but at least we would like to see how qualitatively we can reproduce the phenomena. Okay, so this minimal model, with some quotes, involves two sets of agents. So the cells, which will be modeled as rigid spheres, and the fibers, which will be modeled as assemblies of cross-linked fiber elements. So you should view a fiber as this collection of small fiber elements. And the fiber elements, to make it very simple, have a fixed length. They are, you know, they have a negligible lateral dimension, so they're really like rods, and they have the possibility to cross-link, okay, so when they intersect, this is a 2D model, when they intersect, they have the possibility to cross-link, but not all of them, not necessarily all of them. So this will be, and when they cross-link, there is kind of a torque that force crossing fibers to align, but of course, if you push on this assembly of cross-linked fibers, they will form a kind of angle, and this torque will resist this pressure and will provide the resistance to bending that we like to have. And we also are going to have the possibility that the cross-links actually are formed during the dynamics or are removed. Okay, so that's supposed to describe ECM remodeling during the process. Okay. Okay, so that's basically the main ingredients of the model, and now let's go in more details of the model. So again, the agents are cells, so we're in 2D, so there are disks, their position is defined by xi, the radius ri. And we have these fiber elements, which are just segments of unit length L or LF, and centered at a point which we will denote by yk, and the unit direction omega k. So this is a vector of norm 1, or if you prefer, you can use an angle to represent that vector. This is an angle of line, so it's actually omega k or minus omega k are indistinguishable. Okay, so it's speaking of nematic systems. And a fiber is going to be a set of connected fiber elements. Okay. Now, our model is a discrete in time model with a flowing flow cart. We assume that these cells and fibers are, there are mechanical exchanges, mechanical interactions between these agents. So at any given time, they are in a kind of a mechanical equilibrium. Then there is some disruption of the mechanical equilibrium, which is produced by the biology. That can be cell growth, adipocytes suck in some fat and become a little bit bigger, or cross-links are formed or removed, or other kinds of things like that. Maybe appearance of a new adipocyte. Okay, so there is a disruption of this mechanical equilibrium, and then at the new time step, we are going to try to restore this mechanical equilibrium by minimizing this energy functional, subject to a certain number of constraints that are due to the geometry. Okay, so let's go into more details into these various elements. So we're assuming that two cells cannot overlap. There is volume exclusion. So because they are fat reservoir. Fat is an incompressible liquid. So we are going to assume that when reaching a minimum of the energy, the cells cannot interpenetrate each other, which means that some function of the centers of the two spheres has to stay, say here in this case, negative. So we mean that this distance must stay positive. Okay. The other constraint that we will introduce in the model is the maintenance of fiber length. So whenever we do the minimization of the mechanical energy, we assume a certain topology of the network. So there are some links somewhere, and these links are going to be maintained in the minimization. That means that basically the point on this fiber that links to the point on the other fiber, here I've disconnected the fibers and I've represented these 2 points. These 2 points should be the same. So we are just saying that this point is equal to these points, so that means that some function of these coordinates has to be, and the vectors here have to be zeros. Here LKM and LMK are just the distance between the attachment point on the fiber and the center, okay? So we just have algebraic constraints, so this is an equality constraint, this is an inequality constraint, so we will have to assume that they are satisfied all the time by the algorithm, okay? So that's the constraints. And now the mechanical energy is going to model, so so we can view a fiber element more like, rather than like a physical element of fiber, rather than like of a quantum of fiber resistance. So it means that within the neighborhood of this fiber element, I've displayed some ellipses here, then this fiber element will actually repel the cell. So there is a kind of a repulsion potential between the fiber element and the cell, which has ellipses as level curves. And the other thing that we need to introduce in the mechanical energy is the fiber resistance to bending that's modeled by a torque at the cross-link of 2 crossing fibers that would like, in the absence of any other phenomena, would bring the 2 fibers in an aligned state. Okay, so the torque wants to align the 2 fibers, and we take, a simple formula like this, and this constant is like kind of a flexural modulus of this beam, the fiber of the beam. Okay, so that's basically the mechanical energy, and then we find the total mechanical. We form the total mechanical energy, which is a sum over all pairs of cells and fibers of the repulsion. Fiber cell repulsion energy, and the sum over all connected fiber pairs of the alignment energy. And so we're going to minimize this energy subject to these constraints that I saw, which is basically the non-overlapping of cells and the maintenance of fiber links. And so there are standard minimization algorithms for these kind of things. There's one which is called the Uzawa algorithm that we use, and so there is a little bit of engineering behind that, but this is something that's doable, okay? So now I said that this the step of minimizing the energy functional. So once the energy is minimized, then we are going to introduce biological phenomena that are going to disrupt this mechanical equilibrium. The first one is we assume that when, say, a stem cell differentiates into an adipocyte, that creates a new adipocyte. We do not model the stem cells per se, but once it becomes an adipocyte, we start to model it. So it's a small cell, and we model it as the birth of a new cells. And actually, in order to inseminate these new cells, we have tried 2 different algorithms. One is purely random with uniform probability in the domain. Another one is biasing the appearance of new adipocytes in the location where existing adipocytes are already present. Okay, so here in this case, I would have a high probability to find to inseminate a new adipocyte here or here than anywhere else. Okay, and this is a kind of a way to indirectly model the influence of vasculature, because vasculature brings in the nutrients that's going to favor the development of the adipocytes. So new adipocytes are more likely to be created at the end of the vasculature, which turns out also to be the place where adipocytes are more likely to be. Okay, so there is a correlation between new adipocytes and existing adipocytes, but this correlation is indirectly brought by the vasculature. And one of the things that we found is actually that whether you choose this completely random insemination or this biased insemination has actually little influence on the shapes of the lobules that you find. So it's actually very shocking and striking because there is a kind of traditional thinking is that vasculature is very important to the birth and the development of new adipocytes. And so we will find that actually maybe it's true, but it's not needed because the mechanical interaction is enough actually to bring these the adipocytes together. Okay, so that's one of the other thing that we include is adipocyte growth. So we suppose that there is constant growth until a maximal size is reached. And we also include the creation or the removal of fiber links, which which accounts for, say, for instance, fiber polymerization, cross-linking, or proteolysis if you have the converse phenomenon. So we have some rates of linking and unlinking. And the important thing is that actually the important parameters that we will have to look at is the proportion of cross-linked fiber elements, which is roughly speaking the ratio between the linking to the sum of linking and unlinking probabilities, and one of these two frequencies which shows the unlinking frequency that we can identify as being a kind of ECM remodeling rate. So that's the, okay. So the flow chart revisited is here. So here is the disruption of the biological, the mechanical equilibrium by insemination, growth, or cross-linking, unlinking, and then the restoration of the mechanical equilibrium using cell fiber repulsion, fiber resistance to bending, subject to these constraints, cell-cell non-overlapping, and maintenance of fiber links. Okay, so that's basically the model. So now some results. Okay, so what we found. So typically, so the first thing I would like to show maybe is the most interesting is a few movies. Okay, so in this movie, we are assuming rather, say, low proportion of cross-links, and we're assuming that there is no remodeling of the matrix. So what you see is that, okay, you have actually the regroupment of cells into clusters. You see also that the fiber clusters remain a little bit dense in the sense because as soon as you have like struts in the in the network, then they cannot. I mean, you have rigidity. You cannot remove that rigidity. Okay, so that actually produces this actually not so well organized fiber clusters. Okay. If you keep the remodeling rate 0, so no remodeling, but increase the proportion of fiber links, so if you increase the rigidity in some sense of the matrix, then you can see that this effect of having these dense, very dense fiber kind of clusters is increased. Okay, so that's without any remodeling. And now I introduce that's a case where the proportion of link fiber is moderate, but now there is some remodeling is allowed. So what you see is that the remodeling actually sort of makes fibers much more kind of nice, much more aligned, and still there is a kind of say more moderate directionality in the fiber clusters, a little bit akin of what we've seen on the picture of the experiment. Okay, so but if you increase more the remodeling rate, then you get really highly organized, directionally organized cell clusters, which are not akin to what we've seen on the images. Okay, so this basically these are basically, roughly speaking, 3 phases that we can observe, and which are summarized here. So again, we have here the remodeling rate, low, large. Here we have the proportion of linked fibers, low, large. And so low remodeling rate, and then you have these fiber clusters that are kind of nice. If you increase the remodeling rate, then the fiber clusters are more aligned, and the cell clusters are still a little bit compact. But if you increase again the remodeling rate, then the cell clusters become again very elongated. And so, in fact, we have actually defined some statistical indicators of the fiber alignment and the cell cluster elongation, and we've identified some kind of phase separations by looking at these parameters among this phase diagram. Okay, so I'm not going to get into the details, but this is the thing that we've seen. Yeah, so this is basically what we've seen. So in fact, we've compared with data. So these are actually experimental data, experimental images that could be actually segmented for the cells and then later on for the lobules. So after cell and lobule segmentation, this is what we get. And these are examples of simulations that give the same kind of statistical indicators than what we get in the cells. So here we have comparisons of these statistical indicators for the experiments and for the cells. I'm not going to get into the details, but we've seen, for instance, one of the indicators is the elongation of the cell clusters, and that we could compare with these images. Okay, so that's the kind of thing. This image is a large scale. So here I can give you the scale. This is 500 micrometers. So you see a single lobule is here, but you have many. Okay, so this is the kind of. All right. So this is a mouse tissue. Okay. So here is the thing I said about bias. This was so far no bias, random insemination. If we bias the insemination in order to favor the appearance of new adipocytes where existing adipocytes are present, we change little. We change a little bit. You can see that maybe these clusters are a little bit bigger than these ones, and so on. That is not that striking. So biased insemination doesn't seem to have a major role to play in the structure. Obviously, if you increase the bending resistance of the fibers, then you get more elongated structure, fiber structure. So for instance, compare this picture where the flexural modulus is small to that one where it is large. So no surprisingly, you have rigid, very rigid fibers and elongated cell clusters. The fiber element length has actually also an influence, because in fact if it's short, then you get more round, more smooth structures than when it's large, because of course you are sort of discretizing a fiber which is a curve by smaller elements. So this is what you get, but basically you can see the same kind of phases that you have when you increase the remodeling rate. We have actually introduced some noise, but again, noise has a perceivable but not very strong influence. So it's not a noise-driven phenomenon. Yeah. So I'm just going to summarize. So we were interested in the emergent structure of adipose tissue and basically how it can emerge from coupled cell fiber dynamics. Again, our hypothesis is that it emerged from Curie mechanical interactions, the cell-cell non-overlapping, the cell-fiber repulsion, and fiber resistance to bending. So the model shows that it is possible, that biased insemination has little influence, and that shows that actually vasculature is not needed to account for the observation according to the model. I'm not saying that vasculature is not needed at all, but it's just that the model shows that for that structure to appear, it is not needed. And the model can account for different types of morphologies. As I said, there are various phases, and actually much more if you introduce other phenomena. We have introduced apoptosis and other things, and you can create other structures. And it's actually interesting because then we could actually tune the parameters to match the structures to other organs, like muscles and so on, other organisms, like humans instead of mice, other pathologies, fibrosis, and so on. We've not done that, but this would be possible. So we have highlighted some phase transition, that the major parameter is the ECM remodeling rate and the proportion of linked fibers, and these three phases are here. Just to let you know that we have been able to derive a macroscopic model for the ECM alone without the fibers so far, assuming a certain number of scaling assumptions that aren't valid. Of course, when you're trying to do mathematics, it's always away from the set of real life, but you know, it's the nice thing also on mathematics that you can always dream. So there's very fast ECM remodeling rate, strong fiber alignment force, strong angular noise intensity. Here we need angular noise and small fiber length. In this limit, actually we can derive rigorously an equation, a continuum equation for the direction of the preferred anisotropy direction. So here what we prove is that, as said in the previous talks, the angular distribution of the fiber is a probability, but this probability has a given shape. It's actually a von Mises distribution for those who are familiar with that, and that von Mises distribution has a preferred anisotropy direction, which is this theta, and this theta satisfies these nonlinear parabolic equations, and Jan was able to actually show that there is existence of solution for this equation, and she showed also very nice comparisons between the macroscopic model and the microscopic model, showing that actually the macroscopic model is actually doing the job of describing what the microscopic model does. The derivation is actually not so simple. So there is no physical conservations, because this is basically the theta is the parameter of a director of a vector of norm 1. So there is no conservation associated with that. But we have used some recently developed theory of generalized conservation relation, I would say, which we call in kinetic theory generalized collision invariant, that actually allows you to sort of rigorously derive this equation from the mean field equation associated to the particle model. And the last slide is for future work, and that's future work is simple, is 3D, and so this is a segmentation of the lobules. So it's 200 microns made by the team in Toulouse, and so you see that has nothing to do with something regular. There is a very strong anisotropy direction. It's very much elongated. Actually, this is funnily enough, this is the direction of from skin to the muscle. It's not actually along. It's not along the skin. It's actually transverse, so it's skin to the muscle, and has this kind of shape. And yeah, we have plans to apply the same methodology in 3D, but there's a lot of work to do. Okay, so I'm going to stop now, and thank you for your attention.
The radiuses, are you allowing them to kind of like do you use the same radiuses, or do you allow them to vary? Which radiuses of the circles?
The radius of the cells, they grow. So they start very small, and at each time step there is a constant rate of volumic growth. So we assume that the intake of fat, of nutrients by the cell is constant. So that induces a volumic growth that we in 2D, so it's not completely linear, and there is some stochastic variation around this. Okay, so that not all the cells grow, in synchrony, but this is so at a given time, different cells have different sizes. Yeah.
Is the number of fibers constant in the simulation?
Yes, in the simulation, the number of fibers is constant. Yeah.
So your system can go to a certain size, and then it's
Actually, there is not really any repulsion between the fibers, because, as I said, the fiber element is more like a quantum of fiber resistance. So we assume that when you have a lot of fibers that collapse, that means that you have a very strong fiber there. But obviously, the cells cannot occupy because there is non-overlapping. There is a limited number of cells, and so actually we inseminate adipocytes until we reach about when they are mature, so when they have reached their maximal size, they occupy 50% of the volume. so which is close to packing but not completely equal to packing, and we varied that fraction a little bit. I didn't show that. So what we see is, of course, that the structure that you get vary as a function of that number, but the same trends as observed, so that you get the same behavior as the remodeling rate varies, as the number of the proportion of fiber links, and so on. So there is a certain flexibility or with this parameter, but the trends, which I think is important, the trends are the same. So.
In relation to this, I was wondering what is the final size of the lobules in your simulation, and especially the ratio between your cell size and the fiber size and the final lobule size. Does that compare well?
Okay, we did not do this comparison because it's impossible to segment the fibers. So we do not have any idea of what is the actual size of the fibers in the tissue.
No, I realize, but you do have the size of the lobule in your last picture, and the size of the cell you can estimate perhaps.
Yes. So yeah. So in fact, there is a scaling difference between the experiments and the simulations. We have less cells in the lobules than we do have in the experiments. One of the reasons is that, of course, it's and so we are working on that. Actually, the algorithm is a little bit time-consuming, so we were limited in terms of the number of cells and fibers, and we couldn't really achieve the same number as what we observed. But so in this comparison, we try to account for this difference by looking at reference values, which and measured all those quantifiers with respect to this reference values, which we took for like the average of all configuration we could get. So I agree that's a touchy point, but this is the best that we can do.
It could be possible that the process continues. So does the size of the lobule stabilize in your simulation, or does it continue to coarsen the whole process?
So no, once we have, okay, so this is once we have reached a certain time, then so if you record some of the statistical quantifiers that we have used as a function of time, you see that there is a unary evolution, and then so we've run the simulations about twice the time needed for these statistical indicators to stabilize. So there is still probably some changes, but at least the statistical quantifiers that we have looked at, they stabilize.
How much of your results do you think that will be valid in 3D? Because the depletion forces are, I expect.
It's actually depending on how you're going to extend the model in 3D. So there are various ways to extend the model in 3D, and I suppose that's going to depend on the way you do. So I think that there will be the good ways and the bad ways. Problem is that I don't know which one is the good and which one is the bad.
I mean, the depletion forces push these fibers to come together and produce kind of bundles, but
it's not clear if they are going to bundle like
they can have a completely opposite effect in three-dimensional, because they can push them away from each other, because that extra dimension can let them to get away from.
So the least that we can say is that there will be more effects, and so probably the variety of morphologies and phenomena that we have to put in is certainly very interesting. So this is, I think, challenging. But right now, so I can say that it's not a trivial extension, that there are several ways to do it, that certainly the result will depend on how we do it, and I think that's a fascinating field to look at.
Related question, but on the experimental side. So lobules are 3D structures with surfaces separating them?
Mm-hmm. Yeah. So this is basically, this last picture is is a segmented. So they cut slices with, and then they reconstruct with an imaging software. And this is the kind of, so there is some. There is some segmentation that is done layer by layer, and then you get this. So this is a typical structure, but it's to have in mind that actually the lobular structure is very homogeneous in the tissue. So for instance, this is a slice of tissue that goes from, say, from here to here in the mouse, and is according to the location. Then you may have lobules that are more compact, more elongated, or sometimes you may have actually not really well distinguished lobules. You have a kind of a mixture of the fibers and the cells that doesn't enable you to really segment the lobules. So, yeah. And so actually one of the issues with the continuum model is actually to be able to model the whole organ. And certainly with these techniques, that's not possible because it's beyond the computational possibilities.
Another small comment on the modeling. When you change the fiber element length, the results changed pretty much. Shouldn't there be some kind of a scaling relation, maybe between the bending stiffness and the length, such that this doesn't change, right? I mean, you don't really want that.
Yes, I've not explored that, but I'm sure there is. Yeah, for sure, it's an interesting direction. There's also something that could be added would be some kind of distribution of fiber lengths. So you could try to think what's this. There is some randomness in the fiber length. Maybe it would affect the result. I don't know. We have not explored yet.
Okay, I think we should stop here. Thank you.